You can come to the first slide, Rizvi sir.
Done, ma'am.
Yeah. So, good evening, everyone. Welcome to the 24th webinar of Amity International Society of Natural Products. Today, we are very fortunate and we are very proud to welcome Professor Syed A. Rizvi from College of Pharmacy, Nova Southeastern University, Florida, USA. So, on behalf of our Honorable Founder President, Sir Dr. Ashok K. Chauhan, our Chancellor, Sir Dr. Atul Chauhan, Sir, our Vice Chancellor, Ma'am, Dr. Balwinder Shukla, Ma'am, President, ASTIF, Amity Science Technology and Innovation Foundation, Dr. W. Selva Murthy, Assistant Vice President, ASTIF, Group Captain Kapil Shukla, Sir, and also Director Outcome. From DG Amity Food and Agriculture Foundation, Dr. Nutan Kaushik, Ma'am. From myself, I'm Dr. Harsha Karkwal, and all the scientific fraternity, the students who have joined us, students and scientists who have joined us not only from India, from all over the world. We heartily welcome you, sir. And before requesting sir to begin his webinar, which we are eagerly waiting to hear from him, I'll just read out the brief resume of Professor Rizvi. So Professor Dr. Rizvi pursued an MS and PhD combined degree program at the Center for Biotechnology and Drug Design, Department of Chemistry, Georgia State University, Atlanta. And while working at Georgia State University, he was trained in the areas of analytical chemistry, separation science, bioanalysis, surfactant chemistry, chiral separation of the drug, trace level analysis, that is forensic chemistry of drugs in human body fluids, which led to US and international patent. Right after finishing his MS and PhD at GSU, Dr. Rizvi pursued another PhD at the College of Pharmacy and Health Sciences, Department of Pharmaceutical Science, Mercer University, Atlanta. At Mercer, he was trained in the areas of solid dosage formation, that is direct compression tablet, controlled release formation, nanoparticulate system for non-invasive real-time in vivo imaging, cell-lipid interaction studies. At Renovo, Dr. Rizvi was trained in the area of solid-state chemistry of the drugs, example like polymorphism and co-crystallization. And moreover, Dr. Rizvi pursued part-time postdoctoral research in the laboratory of Dr. Frederick Charles Howard Candler Professor at the Sanford Atwood Chemistry Center, Emory University, where he polished his skills in organic synthesis, soft material chemistry, surface and interfacial sciences, gene transfection studies using Gemini surfactant, state-of-the-art Langmuir-Adam balance studies. Dr. Rizvi has published over 172 peer-reviewed articles, including 110 journal publications, 4 book chapters, 2 books, 1 US patents, and many oral presentations with H-index of 26 and ITIN of 45. His publication includes highly rated articles and 2 cover articles, and 1 figure from Dr. Rizvi's paper was included in the textbook of Qualitative Chemical Analysis by Daniel Harris. He's also achieved the following patent in the research career, which was with Rizvi and Shamsi assay, that is polymeric sulfated surfactants for capillary electrophoresis and CE mass spectroscopy, US patent which was issued on 2012. He presented his work through oral and poster presentations in national and international conferences. He also serves on the editorial boards and panels for 78 different journals and review manuscripts and books for 126 well-known scientific publications. He enjoys teaching and engaging students in thought process. His research interest includes pharmaceutical formulations, ionic liquid formulation, and co-crystallization of pharmaceutical compounds, nano and micro encapsulation of the drugs for targeting and controlled release, clinical and translational research, multi-step synthesis of small organic molecules, pharmaceutical analysis, design and synthesis of novel monomeric and polymeric surfactants, and separation of chiral and achiral compounds, and micellar electrokinetic chromatography, method development and validation for separation of achiral and chiral compounds using MEC UVMS and HPLC UVMS, application of cryogenic high resolution scanning electron microscopy and analytical ultracentrifuge for characterization of polymeric surfactants, chemical transformation of pharmacologically active compounds using microbial whole cultures, isolation and pharmacology of compounds from fungal metabolites and medicinal plants. So, with this vast experience and rich experience, I take it as a privilege to proudly welcome Professor Rizvi to to the webinar of Amity University. So we welcome you, sir, and we welcome you to kindly begin your webinar. Thank you so much, sir. Thank you for
Well,
joining us.
thank you, Dr. Hersha, for the invitation, and thank you to all for joining, and Amity University administration for this opportunity. So, Dr. Hersha has already introduced me, so I'll skip to that slide. So I actually included this cartoon. Actually, it's both funny and a real story. So there's actually a child asking his mom why this drug will get up, from there up to the sore throat. So when I was a kid, I had tonsillitis problems. So, and as a kid, I was afraid to swallow pills. And then whenever I get injection, I used to ask my dad, how's that I'm getting shot in the butt? And then while my tonsils are hurting. So, again, as an adult, if you have a choice of taking an injection or taking a pill, I think most of us would agree that, most of us would not want to get poked by a needle. And then, taking drug from oral route would be actually a professional way of taking a drug. So again, I also do clinical and translational research beside chemistry and pharmacy. So oral route of drug administration, if you search, so I think, I've read different numbers from 80 to 95% prescriptions are for oral administration of the drug for common reason. I mean, it's most convenient. It's usually the safest. Safest, it's just a relative term. If you take, like, say, overdose, you can induce vomiting, and then it's much easier, relatively easier to reverse the unwanted outcome compared to injecting drug into the body, especially IV. It's pretty much done deal. If you make a mistake, you just have to ride that out. So, and then also making solid formulations tend to be a lot more economical, least expensive compared to making an IV formulation or a parenteral formulation. In that case, you have to make drug or formulation sterile and then isotonic and all that. So oral route, tend to be a lot more economical, a lot more, financial incentive as well. And then it's, most commonly used as well. So, but there is a catch. Just like if you take a look at this picture again, so, we just like, in this case, injection was, given in the, butt, and then drug will travel and then, exert its pharmacological effect at the target site. So when you take drug orally, it still has to go through, multiple, pathways. And I've actually simplified this in this figure. So let's assume that you have actually taken a tablet orally. It has to first dissolve in your, it has to first disintegrate. So just like taking a pill, it has to first, become in the powder form. That will happen in your stomach. And the next major step that's supposed to happen is the solubility, okay? And then once drug is dissolved in the intestinal fluid, then drug will become available to pass through the cell membrane and gets into the systemic circulation. So, I mean, unless, your formulation is bad that, tablet will not disintegrate, then you will just pass it out in your stool. But, if the formulation is done right, and that's the case, so tablet has to, there are two steps that can actually hamper drug reaching into your systemic circulation. Number one is the solution, right? So if drug doesn't dissolve, you're not gonna have drug molecule available to pass through the cell membrane. The second step is, after the drug dissolution, is the permeation of the drug from the cell membrane, right? So those are the two factors that can actually control drug reaching into your systemic circulation. So basically, drug absorption from the oral route is solubility limited as well as permeability limited. Now, keeping those two things in mind, well, again, I will show you that slide later on. So, but for most part, the intestinal permeation is usually passive diffusion. That means drug will just go from higher concentration zone to lower concentration zone and reach into the systemic circulation. But in certain cases, there are certain drugs, and then most nutrients will actually need a transporter, a receptor for drug to be captured and then transport them across. So I hope you can see the mouse from your side, that drug will be actually bound to a receptor and will be transported across the cell membrane, and then will get into the bloodstream. So going back to this slide, keeping solubility and permeability in mind, drugs have been classified traditionally into 4 classes: class 1, class 2, class 3, and class 4. Now, class 1 drug is ideal drug. That's where you call hitting the jackpot. And drug is highly soluble as well as highly permeable. So in that case, if you have that kind of good drug, just formulated the way you want it, you can have, immediate release, control release, or enteric coated, whatever you need to. Now, the second class is low solubility, high permeability. Now, again, I've read different numbers, but I agree to a number that about 40% of the drugs that are discovered in medicinal chemistry group in industry and also made in the labs fall under class 2 category, that they suffer from low solubility, but they usually do not have much of an issue for permeability. And this is why actually I put this brain emoji here, so you can actually think there are many tools available for us as chemists and pharmaceutical scientists that we can actually work with it. Now, class 3 is the opposite of class 2. In this case, you have high solubility but low permeability. And class 4 is both low permeability and low solubility. And then this is where you have to think a lot. It's not easy to formulate them. And just because of this formulation factor, drugs which fall under class 4 category tend to be a lot more expensive. Example, bifungizome, it's amphotericin B. It's a lipid formulation. It's actually pretty expensive. Even one dose, I think, can cost up to several $1,000 if I'm not wrong. So for my research, I try to select class 2. Now, class 3 and class 4, you can use very similar approach using, self-emulsification system and then lipid nanocarriers. But as I said, majority of the drugs will fall under or have fallen under class 2 categories. So, and it's very, high, incidence rate of, new molecule, new chemical entities falling under class 2. So I've actually, mostly worked with class 2 compound, biopharmaceutical class 2 compounds. So these are the approaches as a pharmaceutical scientist would consider. Now, again, this is not a comprehensive list, just a generalized overview. So we can actually use carrier method, right? So we have liposome. Nanoparticle is actually very big nowadays, right? So as I was mentioning, liposome you can actually use for class 4 and class 3 drugs. And then you can actually have complexation using cyclodextrin. You can actually do, structure modification. That would be medicinal chemistry, sol formation and emulsification. Now, as a chemist, my first PhD was in chemistry. So, I'm always interested in organic synthesis. So synthetic chemical approaches would involve reducing the log P hydrophobicity. So remember, if drug is not water soluble, more than likely it's very hydrophobic or lipophilic. So what we can do as a medicinal chemist, we can reduce ionizable groups like carboxylic acid, sulfate group when possible. Other polar groups like hydrox group, amine group, hydrogen bond, acceptors and donors. But again, with synthetic chemistry, you have to be careful. You don't want to make molecule too different, then will become what we call new chemical entity, right? And then FDA will actually make you go through the whole, clinical trial. It becomes very expensive. And also you want to preserve the pharmacological activity of your drug molecule. So other way of, making molecule more water soluble is, in this case where we're dealing with a crystalline compound, is to reduce crystal packing. So, and this was highlighted by research that was done by Frey et al. That was published in 2001 in Medicinal Chemistry Journal. So they were actually working with a compound, they're NMDA receptor agonist. So you'll notice the molecule on the left where you see the mouse, the solubility is very low. And when they substitute hydrogen with an ethyl group, it was, solubility increased 100 times. So went from less than 10 microgram per ml to 1 milligram per ml. So again, I was surprised because ethyl group is more hydrophobic, but what was happening in case of this first molecule on the left, there was a lot stronger crystal packing, And if you recall from your basic chemistry courses, some ionic compounds do not dissolve in water. The reason being is there's a very strong interaction between the ions, and the crystal light is so strong that water is unable to permeate and then cause the dissolution. So we can actually use the same principle right here. If we can disrupt the crystal packing, in this case, we are actually using synthetic chemistry approach. We actually had ethyl group was introduced, and we were able to prevent the crystal packing, and then it become more soluble. So co-crystallization actually uses the same principle. So again, in this blue hexagon, you have API, the drug active pharmaceutical ingredient, and then you have guest molecule. So when you make co-crystals, so we're hoping that API will actually talk, so to speak, and then that talking mechanism is via hydrogen bonding or hydrophobic interaction. So that can actually form a co-crystal. It's actually another crystal, and you will notice that the API molecules are separated by coformer. And then we pick coformer such that it is more water soluble, because again, my goal in this research was to increase the water solubility and thus increase the bioavailability. So you'll notice that when this co-crystal hits the water, it actually, the coformer dissolves right away, and then you're left with molecular dispersion of the drug, and that, that makes it water soluble. So there are, so I just want you to pay attention to the vocabulary. So the definition of co-crystal is controversial. You will see different papers, and FDA has actually revised its recommendation a few times. So every year, when we have, ACS meeting or International Union of Crystallographers, when they meet, they just, so I recall from a graduate student in 2006 to this point, 14 years, I mean, this definition of co-crystal has revised a few times. So again, this definition is agreed upon as of now, but it may change. So we define co-crystals as solids that are crystalline, single-phase material composed of two or more different molecular and or ionic compounds in a stoichiometric ratio. Remember, in order to have a co-crystal, it's not an accident. It's a defined structure. You need to have a stoichiometric ratio, right? So it's 1:1, 1:2. And it's again solid and crystalline phase, and it has to have two or more component, right? So that we call co-crystal. So that actually took out the, hydrate. People were arguing, can we call hydrate as a co-crystal? No, not really. So, and then you have API as your drug, right? And then guest or co-former, it's actually, I mean, again, I've actually have generally in, in quotation mark, it doesn't have to be. I mean, I've actually done it myself. You can actually take two drugs and make a co-crystal out of it. Nothing is stopping you. But in general, in order to keep things simple, you want to start with non-drug chemical present and then actually combine them together. So again, if you take a look at this picture back, so again, this is your API, and then your co-crystal is usually a neutral molecule, edible molecule, and you will see them as you move along. I'll present, multiple examples of them. So again, this slide shows you different varieties of, crystal structure. So again, number one is your homomeric, your just simple crystalline drug. And this can be hydrated. We know many crystal hydrates. And co-crystal, this is what I'm interested in. You have your drug and then usually a neutral guest. But co-crystal can be hydrated, that's okay. I mean, as long as we have crystal structure where we can see, co-crystal, having some water molecules in the crystal lattice, it's fine. And then you can have a salt, right? And then you can have salt hydrate, but you can also make co-crystal of a salt by, again, introducing a neutral guest. And one would argue, why would I want to make co-crystal of an ionic compound? So as I mentioned, certain ionic compounds lack water solubility because of their strong crystal packing. So you can actually do that. And also it's an interesting, material. Again, I'll share some examples. There was a tableting issue. So you can make co-crystal and actually you can have better compressibility of the powder. So co-crystallization offers a unique way of making new and advanced material. And you can actually, just like you have hydrated co-crystal, you can actually have salt hydrated co-crystal. And by the way, guys, I actually see a chat box open as well. Let me know if you can hear me clearly. I mean, I'd be happy to answer any question. I don't mind.
Yeah, you are audible, and I've requested everyone, Professor Syed, to, that will answer all the questions at the end of your talk. So everyone is actually posting their questions also. We'll take
Okay.
all of them after you finish your talk. Yeah.
Understood. Okay. All right. Cool. Thank you. All right. So so these are the class of crystal form. So let's move on. Okay. So where do we actually pick co-former, or we call them guests? So there are several resources. So in the US, we look at the FDA website. So if you actually use food material, so again, there's a list called Everything Added in the Food in the US EFS list. You can actually, again, it's maintained by FDA. So if you pick any of those components, so FDA knows them, they are safe. You don't have to actually demonstrate their safety data to FDA. Same thing, generally recognized as safe, we call them GRAS list. And there was actually a book now, I think it's a new edition came out, Pharmaceutically-Acceptable Counterine. You can actually find a whole list, from that book as well. So why pharmaceutical co-crystal? Again, it's a way of making new forms of API. You can actually work with neutral compounds and salt-based compound. And then for pharmaceutical industry, it's a big incentive that, it's a new and patentable form. So for example, if you talk about making a patent out of aspirin, right, people will laugh at it. I mean, it's such an old molecule, but if you make a new co-crystal, that becomes patentable. So again, even you can actually take an old molecule, make a new co-crystal that becomes patentable. And if it turns out to be, very successful drug, you can, an industry can make a lot of money. And, one can argue, but again, this is actually a way of designing new, solid form using crystal engineering. So crystal engineering is an umbrella term. One can use, knowledge of supramolecular chemistry, packing of, molecules, organic, physical chemistry, and actually design new crystal. And then in some cases you can actually make an ionic liquid. I will not go into much for detail, but they're all interrelated concepts. So co-crystals are a way of altering the properties of the drug API. You can change the solubility. I'm using the word change because I will actually talk to you at the end of my project. Sometimes you can increase the solubility. Sometimes you can actually decrease the solubility. You can actually control. So the cool thing about this research is you can actually tweak the solubility the way you want it. And then you can alter the bioavailability. In my case, I was interested in improving the bioavailability. Water sorption, desorption, some molecules are, very sensitive to having water molecule on them. So you can actually control that as well. Purification. So, if you recall from organic chemistry textbook, people have used brucine salt to actually purify their enantiomers. So you can make co-crystal and use purification technique as well. And you can alter chemical and physical stability. So co-crystal is screened. Now, this is a busy slide, but I would take some time and, explain it to you. So how do we make co-crystal? So before I go into all of the discussion, so here is the deal, like polymorph screening. So polymorphs are, for people who are not in the field, polymorphs are, same, just like we have isomers. So they're same molecule, one molecule, but they pack differently, just like your hand. You can make a fist, right? You can have, fingers closed. So they will be what we call polymorphic structure. So just like when we screen for polymorph, because polymorph tend to have different solubility and different bioavailability phenomena, just like when we screen for polymorph, co-crystal screening is also an empirical exercise. That means you have to actually do the experiment. You can do a lot of prediction, and this was the second project that I'm going to talk about today. Is you can do experiment, and then you can come up with prediction tool. But again, tool is as good as your experiment. You actually have to do the experiment and verify that co-crystal will form or not, okay? But we can, however, that's the cash line, that we can increase the odds by a rational approach to experimental design, because you will see there are literally several thousand co-formers. I mean, do you have time to screen every single co-former with a drug? That's not possible. I mean, we have high throughput screening method. So you can actually do what we call pre-screen. So look at the API, look at the co-former, and look at what you're trying to accomplish, your specific aim. And then you can do smart experimental design and then make co-crystal that will be useful to you. So when we look at the API, we look at the structure, look at the form, symmetry, hydrogen bond acceptors, hydrogen bond donor, hydrogen bond donors, salt forming capabilities, conformation, right? In case of, if you have non-saturated drink structure, they have conformations as well, right? And then we also need to know solubility in multiple solvents. Again, that's part of my second project I'm going to talk about. And then chemical activity and stability as well. And then also for the coformer, since you're bringing a non-API in general, you make sure that you know the toxicological property. But if you're using a food product, so it's generally, you know, it's recognized as safe. And again, very important, it has to be pharmaceutically acceptable. So what are the techniques that we use? In the lab, we use thermal method, grinding, and sonication. Now in industry, we actually use grinding method on the larger scale to make co-crystals. Okay? So then you generate your samples, and then you analyze them, and then you scale up. And then there are several drugs out there in the market that are co-crystal. Entresto is one of them. It's very commonly used to people with a heart condition. So there are several co-crystal drugs in the market, and I was actually checking this morning just to make sure that I had the right statistics. So 4 drugs have just completed their clinical trial, and hopefully they will make it to the market. So again, it still is an active area of research. So this is how it looks like. I mean, I wish there was a better picture, but again, the reason why it fades away is just to show you that you can, think about when I was a graduate student, still now, I think about, okay, well, I have a drug, I know how many coformer I can try. So literally, you can try thousands of coformers, but again, it's not practical. So you do your analysis based on your knowledge of chemistry. Then you can narrow down your research to maybe like 10. Even that is actually a lot depending on your resources, and then how many people are working on a project. So you can actually pick up the most important molecule or the target molecule that you think based on your previous experience as well. Again, this is a science where you get better with experience, just like when you're a new driver versus experienced driver, right? So with experience, you can actually get a pretty good idea. Okay, well, these are molecule will likely to form co-crystal. But then also can actually prevent you from, checking other molecules. But again, that's just a balance that you have to decide, okay, well, how much time do I have and how much, how many molecules I can screen. So you actually combine it with a drug, do your experiment, and hopefully you will have a co-crystal or many co-crystals. Okay. So, common methods. So we have what we call sonication or supersaturated solution or slurry. I mean, this is actually a very fast method. I mean, it has, very high, what we call failure rate, but it's also very high success rate. It's 50-50. So useful complete method, what we call HTS, high throughput screen, and SSCI was a company in, I think it's in Indiana. And my co-advisor in my PhD actually worked with this technology. It's a trademark sonic slurry method. I'll talk about that in a minute. And then grinding is actually an excellent method, but it takes a long time. It's actually much more time consuming. So with sonic slurry, you can have a co-crystal in literally seconds, 30 seconds to a minute. Grinding, you have to grind for at least, 20 minutes to 30 minutes or sometimes even more. Now, melt experiment is called Kofler technique. It's actually a cool method. You can actually see a co-crystal in front of you forming, but again, every compound is not a candidate because in this case, you have to melt the coformer and the drug, and in some cases, the products are heat labile and they will decompose. So this is sonic slurry method. So, this is a sonic probe. I mean, people who work with cell biology know this. We use them to disrupt the cell membrane, lyse the cell, right? And then this is actually a 96 well plate made out of aluminum block. I mean, if you try to do this experiment in that plastic 96 well, I mean, solvent will actually chew through the plastic. And then because we're using very high frequency, that will just kind of, break the well as well. So what we do is we actually label them, and then we take certain amount of drug, we weigh them on paper, add them and add solvent, and just literally give them a sonic buzz. And then the solid that is formed, we can actually analyze them with several techniques, so Raman or X-ray powder diffraction, and you can analyze them. So it's a really cool and really fast method of making co-crystal. So we actually make a slurry, again, where goal is to use solvent as a mediator. Remember, we're not using solvent to dissolve stuff. We're using solvent to bring the coformer and the drug together. It's just actually a medium for them to, talk to each other, so to speak, okay? And can also, initiate primary nucleation. And as I said, it's a really fast and high throughput, a true high throughput method of making co-crystal in the laboratory. So liquid assisted grinding is actually another common technique that people use. I actually have this in my lab. We actually use mixer mill. So we have, those are small well made out of, stainless steel. And in which you have actually grinding balls. You can have 1 or 2, and then you can add powder together and some solvent, and then you grind at very high frequency for a long period of time. And this is why the shield is out there to protect, just in case if this actually gets dislodged. But I mean, this is a time-consuming process, but actually success rate with liquid-assisted grinding is very high. I mean, I would say, over 90 to 95% success rate compared to the liquid-assisted grinding. As I mentioned, it's time-consuming. Now, if you were a graduate student, if you're working by yourself in the lab, 30 minutes waiting, it looks like taking forever, right? Okay. So in the Kofler method, I mean, it's pretty cool. You actually, you melt your API and the guest, and you actually put them on the slide. And then you can actually watch under the polarizing microscope. So this is a guest molecule right here, and this is my API. As they come together and then actually cool down, if they were to form a cocrystal, you can actually see cocrystal forming at the boundary in real time. It's pretty cool. But the problem with that is, if it is a volatile compound or heat labile, they may actually break down, but actually it's really cool to observe. So you can actually use Kofler method, but it's not very popular. It's limited to certain API and the guest molecules. So now let's talk, real stuff. So I'm going to present to you 3 projects. One project was actually a compound called M. It was a sodium channel blocker, right? So, this compound came to us. So it was weakly basic API. It has poor water solubility of free base and was low plasma concentration. Remember, before this drug goes into human trial, you actually have to have, favorable pharmaceutical properties. So our goal was to, evaluate cocrystallization to produce more water soluble form. So the methodology was, so we actually had 23 pharmaceutical acceptable carboxylic acid. Again, if you ask me why 23, again, we use our previous knowledge, previous experience. So we narrowed down our research to those 23 pharmaceutically acceptable carboxylic acid. Now, if you ask me why carboxylic acid, if you look at the structure right there, you see that you have amide bond. So these are hydrogen bond acceptor, donor acceptor. So we have plenty of group that will be, happy to hydrogen bond with carboxylic acid. Again, use your knowledge of chemistry and the structure conformation as well. And then again, we tried solvent composition and crystallization condition with multiple ratios of guest and API. And then concept of each well, again, if you recall this picture, we were using this high throughput. So each well, again, you can do 96 experiment at a given time, so to speak. Were evaluated, right? So if you have Raman spectrometer in your lab, literally you can analyze each well, just take out the solid, dry it on a towel, on a paper towel, and then you can take the Raman and you can get, crude results really quickly. So I mean, literally you can evaluate 96 cocrystal within an hour or 2. And then Raman spectra were examined, and then I use the word possible hits. So once you see that, okay, well, I'm actually getting, cocrystal formation, it may not be very pure, but then actually you can further optimize that. So again, in the high throughput method, you're looking at whether you have cocrystal or not. So you're looking for the hint or hits, and then you can actually further explore that. So we discovered 5 cocrystals: benzoic acid, fumaric, gentisic, and glutaric acid. So glutaric acid cocrystal was selected because it has very high water solubility. Again, where our aim was to increase the water solubility, and hopefully by increasing the water solubility, we'll be able to increase the bioavailability as well. So so this is based on the crystal structure. It's 1-to-1. So you can use NMR, you can use mass spectroscopy or elemental analysis. So you can actually get an idea that, this 1-to-1 ratio between drug and your coformer, right? So, we do the whole 9-yard crystal, characterization. It has to be pure. In certain cases where we make single crystal, we actually have single crystal structure available too. So this is an example of Raman spectrum. This whole thing literally took me less than a minute. So you have your API, your guest, and your cocrystal. So you can actually see, so red one is actually a cocrystal. So you will see the peak will shift. You can, analyze multiple peaks, but I just, look at the area right here. So I can see a very nice API, guest, and cocrystal. So, this shift in the peak will give you an indication. Yes, you have, possible cocrystal, but again, it could be a polymorph too, right? So you have to do further analysis. You do NMR, you do X-ray and whatnot. Again, I'm just showing you Raman to give you an idea. That's a real quick method in the lab that you can do, and then narrow down your research. As I mentioned, even with, I started with 23, ended up with 5. So you can actually do this in high throughput, method and save time. And then ultimately we do extra powder diffraction to further confirm, because Raman may not actually show you that your cocrystal is pure. So again, so this is XRPD, that's actually extra powder diffraction. So the next step would be, to do NMR mass spectroscopy and then single crystal. In some cases where we can actually grow good crystals, we'll do single crystal as well. So again, shift in the peak will actually indicate that you have a successful cocrystal formation, but extra powder diffraction takes a long time and expensive as well. So once we identify and properly characterize our cocrystal product, so the next step was, is it useful? Is it going to be helpful? So it's an industrial collaboration. Some of this work was done in industry. So you will notice that in vitro and in vivo correlation, that's what we're looking for if possible, and FDA really likes it. So if you can predict the bioavailability in the blood by increasing the solubility, so if you can predict what's happening in vitro and can replicate that in vivo, that's called in vitro in vivo correlation. So in our case, so this is our in vitro result. So you can notice that cocrystal hits the water, bam, very high water solubility, and it actually maintained very high solubility compared to the API. And then, so if you look at the time zero, look at the solubility difference, right? So yes, we had a very highly water soluble crystal, but is it going to translate beneficial in vivo? So here we go. Now this was done in dog. Again, that was done in industry. We don't have facilities to do this kind of study with a dog model. So you notice that with the API cocrystal, the area under the curve was increased 4 times. So yes, bioavailability was very high. So actually in our case, this is a perfect in vitro in vivo correlation. So you notice that we were able to increase solubility in vivo, and it did translate in vivo as well. We actually had much higher concentration and very rapid onset as well. So this is an example of a successful project where we actually got a compound that had solubility issues. We made cocrystals, and it doesn't take very long if you have experience working with this technology. And we were able to make a cocrystal with improved water solubility, and it did translate into improved bioavailability. So that was actually a very happy project for us. So with that, let's talk more on philosophical aspects of cocrystallization. So this is the second project I'm going to talk about. The first one, we actually made, showed you how we make cocrystal and then, improve the solubility and work. So the next project, I think we have time. So in this case, we did actually a qualitative research. Okay, is there a way we can actually help predict whether cocrystal will form or not? If you recall from my earlier slides, we're talking about it's being an empirical science. You have to do experiment, but are there ways we can actually cut down our time and improve the outcomes? So yes, so we actually did this research. So the specific aims were to assess the role of solvents in cocrystal formation and quantitatively predict the outcomes. So by doing those experiments, you will see the utility of this approach. It's actually a very highly cited paper. You can actually predict with very high success rate whether your experiment will yield a cocrystal or not. Okay. So in this experiment, we did not work, with new drugs. Those crystals are not even new. I mean, we just picked this as a model system because we know these molecules very well. We know those cocrystals very well. So we're just using, different solvent and different conditions. So, and then we were trying to come up with a model. So I just want to tell you that these are not new cocrystals, but we picked those systems because we can make them reliably, easy to make them, easy to work with. Okay. So we have theophylline and caffeine. They're very closely structured, right? So theophylline has hydrogen group right around this nitrogen. Caffeine has methyl group, right? Malic acid and tartaric acid. So this is the vocabulary. So you have theophylline with L-malic acid, right? So dot means it's actually a cocrystal. And the second one is TP2. There are 2 theophylline molecules. That's in first case, you have 1-to-1 ratio. In the second case, you have 2 drug molecules with 1 coformer, right? So this is how, remember, if you recall from my earlier slides, that in order to have a cocrystal, you're supposed to have a defined stoichiometric ratio. So it could be 1-to-1, 1-to-2, 2-to-1, but it has to have a defined ratio. And then caffeine and then malic acid and tartaric acid form 1-to-1 cocrystal as well. So what we made an assumption that the target cocrystal will be the thermodynamically favored solid state phase under a condition in which both reactants are saturated. So what we did, we actually defined a ratio called eta. So if you recall, the percentages are defined, weight over volume or volume over volume or weight over weight. We actually did opposite because what we're trying to look for is how much solvent we can use. Remember, if you increase the solvent, you can actually increase the dissolution, and that can actually alter the outcome of your experiment. So for this experiment, again, we did not use Kofler method. We used grinding and sonic slurry because, they're rather, faster to work with. So the way we designed the project, first of all, we wanted to determine the solubility, and you will see the utility of it in a minute, in a high throughput manner. And then we did sonic slurry screen. We actually had 3 solvent conditions. So 2, 6, and 12, if you recall again, we're talking about eta as volume of solvent on the higher. So if you want higher number, that means you need to have more solvent or less samples, one way or the other, right? So if you go from 2, 6, and 12, you're using more and more solvent, right? So remember, this is microliter per milligram. Keep that thing in mind. And for liquid assisted grinding, we use, again, we don't want to use too much solvent in the well. So we actually just use 2 extremes, very low solvent and, high amount of solvent. And you will see the reason in a minute. So we did that same experiment. So we had 3 possibilities, such that in each solvent, API and coformer will have similar solubilities. The second possibility is API is more water soluble, or conversely, coformer is more water soluble, okay? So again, I don't want to bother you with this table, but again, just want to give you an idea that yes, I did all those experiments. It took me quite a while, but again, it's worthwhile, especially if you are going to train PhD students or postdocs and undergraduates. It's a good exercise, So we work with different solvent. Now for drug purposes or for a molecule that you plan to actually make them in commercially viable formulation, you want to avoid chlorinated solvent like dichloromethane. But because in our case, we're just doing a qualitative research, we actually pretty much, look at every possible solvent that we could get our hands on. So we use water again, universal solvent. We use methanol, ethyl alcohol, dimethylformamide, organic solvents, acetone, ACE, acetonitrile, and whatnot. So again, we actually determine the solubility milligram per ml for each of the component, both the coformer and the APIs. Again, it's a busy table. I'll actually send you a link of the paper, so don't worry. I will just actually tell you exactly what's happening in the whole table with those 3 examples, okay? So, but because since we have to report, so we have to, this is directly coming from my paper, so we actually report all the results. So as I mentioned, so there are 3 possibilities, right? So you have solubilities of API are very similar, right? The second possibility was API is more soluble. And the third possibility is coformer is more soluble. And you will see those right here. In the first case, right, 20 and 18, not much of a difference. So we're using acetonitrile. And then because they were both 100% saturated, we got Cocrystal. CC stands for Cocrystal in this, okay? In the second example, there's a difference of solubility like twice. So caffeine and malic acid. So malic acid was twice as much soluble compared to caffeine. But again, they both were saturated. So you got the Cocrystal plus an impurity of less soluble product. So in this case, what was less soluble? Caffeine. So you got the Cocrystal, but you also got an impurity of caffeine in this case. So it makes sense because first of all, the system was saturated. So you got the Cocrystal, but because you have less solubility from caffeine, so you actually got this as an impurity, so to speak. And in the other case, if you flip that, in this case, you have very high solubility of, coformer. So such that you did not have saturation for the coformer. In this case, you just had only left with less soluble, theophylline, in this case right here, theophylline, and you did not form any Cocrystal. Again, if you look at the whole table, scour through the whole table, you will notice that when 100% saturation is accomplished for both API and coformer, you always had Cocrystal, okay? If they're equally soluble, then you get pure Cocrystal. But when there is a difference in solubility for either one of them, then you will have impurity. And in a case where you have less than 100% saturation for API or a coformer, you are very unlikely to form Cocrystal. This is actually a very good, proof of concept that if you are going to use a solvent, remember solvent is there to make a slurry, but we also know that solvent can actually dissolve the compound. So you have to be very careful what kind of solvent do you use and how much solvent do you use. So you want to keep saturated conditions, and that will result in, almost guaranteeing the Cocrystal formation. We did the same thing with liquid assisted grinding. So these were coming from sonic slurry where you use a sonic probe. And then when we use liquid assisted grinding, now in case when we had very small amount of solvent, you notice that in each case, API's saturation was 100%, co-former was 100%, guess what? Every time we had cocrystal, right? But if you increase the solvent, when you actually go below 100% saturation for API co-former, you're not going to get cocrystal. So you will notice that when there is actually a mismatch in the percent saturation, your experiments were unsuccessful. So what did we learn? So so again, before I go on the conclusion, so this is what's happening, right? So a solid, so let's say that one is a drug, one is co-former. Let's assume that we're talking about two-component system, and I'm keeping an eye on time as well. So a solid gets into solution, B solid again is in equilibrium, and then in the solution, so they come together to form solid, right? So if you maintain conditions such that you have saturated solution, solid will be the predominant form. But if you have a solid that has more water solubility, in this case, it will not be able to actually come out of the solution, the supersaturated form. So this is the gist of this talk. Well, just of this particular project. And we were actually using exit powder diffraction. Again, I will just quickly go over because I have to talk real quick on the last project. But this is our pure cocrystal. As we notice that if you increase, in this particular case, we systematically increase the eta value, and we're able to actually, make this impurity product disappear, and we actually had a cocrystal. So you will notice that we're using very low amount of solvent, so 0.04 up to 1 microliter per milligram. And same thing with the other cocrystal. And in this particular case, we're looking at the time. So you notice that, if you give more time, you're likely to make cocrystal. So in this project, we learned using eta value, solvent to solid ratio 1:1, such that you can actually maintain 100% saturation for both co-former and API. You're likely to get cocrystal. Again, I will rather quickly go over this. So we actually made a prediction. So where X and Y are the stoichiometric ratio of the component, A and B are the solubility, molecular weight, and A is the solubility. And you can actually come up with an eta value predictor. If you know the solubility, this is why, if you recall, we actually determined the solubility. That was the whole reason. So we can actually use this model. So we actually determine the solubility. So you can actually plug those values in this equation, and then you can actually kind of predict whether you're going to have cocrystal or not. So we noticed that eta value, again, we actually validated this model as well. And so if you are going to use eta value based on prediction, let's say a prediction value comes out to be 1.77 in this case. So if you keep your eta below 1.77, you're likely to get cocrystal. If you increase, that means using more solvent, you're not going to be able to have saturated condition, and your cocrystal will not form. So in this particular project, we learned how to actually use different solvent and control the solubility. And by knowing the solubility, by knowing the saturation status, you can actually very successfully predict the cocrystal formation outcome. So this was published in Crystal Engineering Communications, been cited almost 100 times, very popular paper. So now this is the last project, so only 3 slides, we'll quickly finish. So this is the project where I learned how to be humble, okay? So the first project was very successful and made beautiful cocrystal, increase the solubility, increase the bioavailability. Second project, yes, we made a model. So in this project, I was actually trying to make a formulation. So we already know there were almost over 55 cocrystal of carbamazepine. So we actually choose, so carbamazepine, again, it's a very low solubility molecule. So we're trying to actually make a better formulation. Patient have to take a lot of this drug, and it's it has a lot of side effects. So we're trying to hope to come up with a better formulation than commercially available. So very hydrophobic and poor water solubility. We're trying to improve the bioavailability in vivo. So it's low plasma concentration by drug itself, heavily metabolized, and you need large dose for pharmacological actions and anti-epileptic drugs, so to speak. So, so this is actually commercially available drug called Tegretol, right? So it's a direct compressed tablet because carbamazepine, when it comes in contact with water, forms dihydrate solvate, and it's actually 10 times less soluble. So carbamazepine is already very poorly water soluble. When it makes a dihydrate, it becomes even less soluble. So that was not ideal. So people that are actually making a direct compressed tablet, we don't use granulation, so people who have pharmaceutical background. So in our case, we actually compare commercially available formulation with our formulation. We actually mimic, right? So we use CBZ equivalent. We use microcrystalline cellulose, sodium carboxymethyl cellulose, magnesium citrate, and aerosol. So, and then we actually made direct compressed tablet. So once you made the tablet, before you do, tablet hardness tester and then the whole pharmaceutical 9-yard, we were actually doing intrinsic dissolution. So intrinsic dissolution, you actually do what we call surface dissolution. It's actually very commonly used, and you can actually do a multiple, characterization together. So what I mean by that is right here. So while you're doing your dissolution, you can actually use a Raman spectrometer to actually see what's happening on the surface. You can use UV to see what's going on in terms of concentration, whether you're getting more product or not. So you can do multiple things with this intrinsic dissolution. It's a very useful technique. So your tablet is entrapped. Again, this is just a drawing. So tablet dissolving from the surface. So if you call your USB dissolution tester, you have a tablet in the sink situation, right? And then you have paddle rotating. In this case, we're just having a stir bar at the bottom, and we're actually letting solvent move from the surface. And you can actually keep an eye on the surface as well. So this is what happened. Okay, so again, I apologize, this is a crude curve, but I just wanted to show you. So in our case, so since we're doing what we call UV detections in real time, so you notice that I was hoping that this would be my co-crystal, right? So bam, higher solubility, and then we'll continue to go up just like we saw in the first project. But to my surprise, co-crystal was the least soluble, and CBC has higher solubility. So I thought maybe I made a mistake. I repeat this experiment hundreds of times. And I was surprised. I said, oh my God, before I call it a failure, I tried this like, I'm an analytical chemist by training as well. So you can imagine how many times I repeated the experiment. So what was happening, if you recall, you can actually do a surface characterization as well in real time. So this is exactly what was happening. So co-crystal was so water soluble. As soon as it hit water, it started converting into dihydrated form. So that was 10 times less soluble. So you notice that co-crystal hits the water, bam, right there. It actually converts into less soluble dihydrated form. So in this case, what I was actually seeing is a dissolution of CBC dihydrate, not the carbamazepine by itself. So as I told you that carbamazepine is already very less water soluble. And then when you make dihydrate, it's even less water soluble. So in my case, making a co-crystal was actually counterproductive, right? So this is the last slide, and before I show you the conclusion. So just remember, co-crystal can be beneficial, and sometimes it may not offer you any, particular advantage. But then again, there's another thing called, parachute effect. Again, I don't want to go into too much of a detail. You can actually use some formulation tools to actually kind of slow this conversion down. But in my case, I mean, I tried several approaches, but it didn't work out. And then I realized, it's really not beneficial to spend too much time on it. But again, this actually, made me humble. I thought that co-crystal can solve every problem. It is not the case. Remember, just like every aspect of science, you have success and you have failure, but they all teach you a lesson, okay? All right, so the conclusion is aqueous solubility of API is very important, right? And then majority of the cases does dictate bioavailability. Remember, if you're taking drug in the oral form, if it doesn't dissolve in your intestinal fluid, you're not going to have drug, in your systemic circulation, right? So many methods are available. So some of you are in pharmaceutical industry and in the field. You can use medicinal chemistry techniques, but try not to change the structure too much. And then you can use formulation techniques like what we call complex formation and the nanoparticle and all that. So Cocrystal is an excellent opportunity. It actually allows you to use your chemistry knowledge, solid state chemistry. I mean, it's an excellent training program for young scientists. Crystallization is easy. Well, easy is a relative term. It's not easy to begin with, to be honest with you, but it's rather safe and cost effective and quick method to generate new solid, right? And then it's important to choose the right co-crystal. So as I showed you from my last example, I had a co-crystal that was 150 times more soluble than the drug itself, but when as soon as it hit water, it converted into a different form. It can convert into a polymorph as well. And that can actually take away the benefits. And I just looked at literally about an hour ago at the clinical trial website. So there are 4 co-crystallized drugs. They just completed their trial and ready to hit the market. So again, none of this was possible without the collaboration and guidance. So Professor Menger is my postdoc advisor at Emory. Dr. Scott Childs, he actually the one who introduced me to the field of co-crystallization. Professor Robin D. Rogers, distinguished professor. He was at University of Alabama, went to McGill, collaborator. And then Alan Myerson at MIT, and then funding from different funding sources. So here we go. Keep calm and grow co-crystals. So at this point, I'll be happy to take your questions.
Yeah. So we are, thank you so much, Professor Rizvi. And I think I saw through all the chats and everyone is thanking us from India and around the world for a very, very informative webinar that you have given. And they're asking also for the recording. So I told them we'll be providing recording in 2 weeks' time. We are joined in the panel as I had welcomed you on behalf of Group Captain Shukla sir, who is Assistant Vice President, and also, he's the advisor to our Honorable Founder President sir, and also Assistant Vice President at Amity Science, Technology and Innovation Foundation. We are also joined by DG Amity Food and Agriculture Foundation, Dr. Nutan Kaushik ma'am. So I think because there are many questions, shall I take questions first, 3, 4, and then
yes, you
that
take
will be better.
Yeah, continue with.
Okay, so I will take few questions because there are almost 20 questions, Professor Rizvi. So, and I think they are still coming in, and 28 chats. Everyone is telling like not only 20, I think more than 50, because I was continuously going through the chats. So the first question is from Dr. Manish Kumar. He's asking, sir, because you belong to medical background, have you prepared any drug which can which can target like SARS COVID thing?
Yeah, I have actually published 2 papers on COVID. My 3rd paper is under review, and I'm working on a 4th paper. So because this is a new viral disease, but we have some understanding from previous outbreaks. So coronavirus is not new. I mean, there are many strains out there. So the first outbreak of SARS, severe acute respiratory syndrome in early 2000, and then MERS, Militant respiratory syndrome, that was in Middle East. They were all coronaviruses. So we have some understanding in the past. Now this new virus, because it actually causes cytokine storm, so we have actually better understanding. So again, there are some antivirals people have tried, and for this particular case, I looked at literally hundreds of papers. So there are several treatment protocols depending on the stage of disease. So early on, you're actually trying to prevent the cytokine storm, prevent inflammation, and your goal should be to prevent patient going on ventilators, because going on ventilation, I mean, you have to sedate them. Unfortunately, if you notice from around the world, people who went on ventilator, very few survived. So we have recently used dexamethasone, some antiviral drugs as well. So we actually had some success. And then there are some antivirals out there in the market that have been tried on. So hopefully we'll take care of this problem in short period of time.
Okay, and they're asking like this is from like one of the he's from Dr. Ishrat. She's asking like, are there transporters on the plasma membranes for transporting the drug that have the low solubility?
So that will be dependent on, remember, cell membrane has, a finite number of receptors.
Yeah.
So yes, if a drug is like actively transport, again, remember, the goal of this is to make drug more water soluble. So if drug is not water soluble, it will not reach to the cell surface anyway, right? So the whole point is to actually increase the solubility. Whether you have transporter or not, that's a secondary issue. The first issue is, if you cannot make drug molecule talk with the receptor, because they don't even see it, they're just like literally a block passing through your intestine, right? So if the drug molecule will not at the molecular level talk with the receptor, remember, if they're not soluble, they're just like clumps, right? So receptor will not talk with the clump. So in order for drug to talk, so to speak, they have to be solubilized. So this project was to increase the solubility. So if they have receptor, that's good. So I mean, many drugs will actually go through receptor-mediated transport, like G protein coupled receptors and whatnot. Yeah. But the whole project at the beginning was to increase the solubility.
Okay. And the next question is from Drisha. She's asking, is it possible to increase the solubility of phenolic compounds by co-crystallization approach?
Yes, very easy. I have actually done that.
Okay. And other question is, what is good idea molar ratio for APIs, active pharmaceutical ingredients, and conformer for a good and efficient co-crystals?
We start with 1:1. So I have actually noticed that we always start with 1:1 molar ratio. And then if you notice that, for example, in case of thioflin, it was 1:2, you will notice that you have more drug left or more coformer left. So then you can actually alter the ratio later on. So if you don't know, start with 1:1.
Okay. And can we make cinnamon more soluble orally through this technique?
So you mean the active ingredient in the cinnamon? Yes.
Yeah.
So you can do that. Sure. I mean, because it's cinnamon, I actually bought the cinnamon oil actually recently. I mean, I do oil diffusion, it just kind of, calms me down. So you can actually, there are many other techniques. So yes, you can use Cocrystallization. There's an ionic liquid formulation. You can actually use what we call self-emulsification technique, because if you're taking it orally, there are several ways out there. Cocrystallization is actually a lot more expensive. I mean, there are a lot cheaper ways out there. Yes, you can technically, yeah.
Okay. And sir, like this is from Dr. Chandravali. She's asking, sir, in short, which factors affect the solubility of drug?
So the Lipinski has a rule of 5. So, I mean, you have to have a certain molecular weight. So if molecular weight is between 200 and 500, more likely to water soluble. And then depend on their, if it's a crystalline substance, if the crystal packing is so very strong, it will have less solubility. On amorphous drug tend to have higher solubility. And then having ionic groups, right? Sulfate, carboxylic acid, and then hydrogen bond acceptors and donors, they're likely to increase the chances of having a compound that is more water soluble.
Okay. And they're asking like some of the herbal extracts will be in sticky form. So do we apply co-crystallization method to overcome this difficulty?
You mean the isolation of natural products?
Yeah.
Well, yes. So, I mean, if you recall, I'm sure we have all taken organic chemistry 1 and 2. So there was actually brucine was used in the past to actually isolate enantiomer compound. So you can actually make a crystal of, an enantiomer compound, and actually you can isolate them. So yes, in certain cases, you can actually make co-crystal. But again, the problem with that would be, I actually had masters with natural product chemistry. I mean, they're already in teeny tiny amount, right? So making a crystal to purify them will be very difficult, because see, if you're isolating a new compound, you don't know what you have, right? So
correct.
Isolating a compound, and then you have to do like 2D, 3D NMR, and if you can go crystal, I mean, isolation of natural product, it's very difficult. I remember I did my master's thesis on it. In 2 years, I was going crazy.
So my student could isolate 9, novel compounds during her PhD. One of my students.
Oh my God! Yeah, so, hats off to her.
Hats off to her, seriously. And this is from Zainab. She's asking what benefits you found in pharmaceutical chemistry using co-crystals over other state of solutions like crystal, solution, and other forms.
So co-crystallization is one method. There are many other ways you can increase the solubility. So in this talk, I was focusing on co-crystals. So when you use the knowledge of supramolecular chemistry, you can actually make ionic liquids. You can make different polymorph. You don't have to make co-crystal. So again, in industry, your job is to maximize the productivity, spend less amount of money, and make the more benefit, right? So if nothing else works, right? So this is another technique, another option available to you. And then for students, I mean, when I was, PhD student myself, so it was a great training opportunity. You come to learn X-ray crystallography, you learn Raman spectroscopy, and then you learn formulation, you do organic chemistry. I mean, it's a multi, specialty tool in one. So you learn so much from it. So my first PhD was in analytical, organic chemistry. I was making surfactant for cal separation. Now, practically industry, you don't use that. But it was a great training tool.
Correct.
Yeah. So it's one of the options among many.
Among many. And we have more than 100 questions, Dr.
I'll be happy to answer really quickly.
No. So because of paucity of time, I request you, can I share your mail ID with everyone in the group? And
Sure.
In LinkedIn and in Facebook, we have Amity Herbal and Natural Product group. So in that, I'll be sharing sir's mail ID. So you can directly post. Otherwise, you have my mail ID and you can directly send me questions. And I'll request sir, whenever he gets time, he can answer all those questions. And now I request, and we are also grateful that Dr. Rizvi has got many, $1 million project, and he's ready to collaborate with Amity University. And we request our panelists now, Group Captain Kapil Shukla sir and Nutan Kaushik ma'am, to please say a few words.
I would request Dr. Nutan Kaushik to speak first.
Ma'am, Ma'am, are you audible? Ma'am, kindly unmute yourself, ma'am.
Hello,
Yeah.
can you
Yes, ma'am. Yes,
is
ma'am.
am I audible?
Yes, ma'am.
Yeah.
Yes, ma'am.
Thank you. Thank you, Harsha, for arranging such a wonderful lecture. First of all, I would like to apologize, Dr. Rizvi, for not joining early. I was having some problem in joining, so I missed such an important and such an informative lecture today. The presentation, it was such a wonderful, because I'm also a bit of a natural product chemist, analytical come natural product chemist. So we do isolate a lot of compounds, and we do face a lot of problem in the purity, getting the pure compounds from the mixtures of the compounds when we isolate from either from the microbes or from the plants. So it's wonderful that Dr. Rizvi is there to collaborate with us, so that our students can go and then learn that these techniques, the
Ma'am, you're not audible.
I think yeah, we lost the audio.
Yeah, we lost the audio, I think. Ma'am, you're not audible, Nutan ma'am. Can you hear me, ma'am? So I request Group Captain Kapil Shukla sir, and then we'll again come back to Nutan ma'am. Yeah.
Can you hear me?
Yes, sir. Yes,
Dr.
sir.
Okay. Thank you, Dr. Rizvi, for a very informative and interesting talk. And on behalf of Honorable Founder President, Sir, the Chancellor, and the Vice Chancellor, I would like to thank you. And I also thank Dr. Kharkwal, Harsha Kharkwal, so that she has brought you here, and so many people have the opportunity to listen to you.
Yeah.
And I hope that in future we will work together with Amity. Once you are with Amity, you are a part of the enlarged family, and we look forward to working with you. And thank you very much. And you have given us hope that we will soon be able to have medication for COVID-19,
Yes.
which the whole world is fighting. Thank you so much, Dr. Rizvi.
Yeah.
Thank you.
Thank you so much, sir.
Thank you, sir. And ma'am, are you audible now? Can you again rejoin if you want? Like, you can try once before we finally close.
I think her audio and now she's there.
Now
Yes.
she's there. Yes, ma'am. Ma'am, once you speak, if you are audible, we'll just see.
Speak out, Dr. Nutan Kaushik. Is your audio working, Dr. Kaushik?
I don't think so.
I don't think so.
Yeah.
Okay.
So, Dr. Rizvi, as you said, like you have got so many projects, and because of lockdown, you're stuck, but we would like to welcome you to Amity after the lockdown, and definitely we look forward for collaborative projects with you, and also student and faculty exchange, as sir told. So, because we have many Indo-US openings, so definitely, and we have got a very strong pharmacy group